So like I said, we are going to be repeating yesterday's first video, which is to make sense of the terminology, because it was a little bit at the last minute and I don't want, oops, sorry. Okay, I'm assuming you can all see this. So DNA is double-stranded and RNA polymerase transcribes only one of the two strands. So the terminology needs to be described very well. So here we have, these are the nucleotides that are coming in. This is, for example, ATP that is coming in. Everybody can see this, right?
Yes, Ajahn.
Okay. All right. So what we want to understand is exactly how the process is occurring. And so this is, for example, the nucleotide, for example, ATP, GTP, UTP. These are all, actually ATP can't come, UTP. These are all coming and they are forming a phosphodiester bond in the 5 to 3' direction that we have learned before. So the phosphodiester bond that happens, that is formed in DNA, the same phosphodiester bond is formed in the RNA molecule as well. The exceptions, as you know, are the presence of the ribose, the presence of the absence of adenine, we just have uracil here. And of course, we need to understand how this process, where the energy for this process comes from. So how is this phosphodiester bond formation catalyzed? The energy for this formation of the phosphodiester bond comes from the inorganic pyrophosphate that is generated. So let's say GTP is being incorporated. The GTP will be incorporated in such a way that one of the phosphates will remain and two of the phosphates will be released. So remember, in the very initial weeks, we talked about alpha, beta, and gamma phosphates, the three phosphates on the nucleotide. Alpha and beta phosphates are released together as PPI, pyrophosphate, and the gamma phosphate remains on the nucleotide now, well, the nucleotide with the phosphates removed, and that gamma phosphate is involved in the formation of the phosphodiester bond with the 3' OH of the upstream nucleotide. So when this PPI is released, this pyrophosphate is released, this pyrophosphate has a lot of energy in it, and breakdown of the pyrophosphate releases a huge amount of energy, which makes this reaction go in a forward direction. So you do have the phosphodiester bond formed, and you have the elongation of the RNA chain because of the release of energy from the breakdown of the pyrophosphate. This huge release of energy makes this reaction go in the forward direction, as we learned in our bioenergetics lectures in the previous weeks. So let's have a look at how this is working. So the term antisense strand, non-coding strand, and template strand, they all refer to the same strand. So now we are going to try and understand what is the meaning of template strand but non-coding strand. So let's start with this gene sequence. This is a hypothetical gene sequence which has this particular specific sequence. Always we read sequence in a 5 to 3' direction. And so it's the 5' end has the cytosine, and then we have the adenine at the end. This is a gene, right? So this is not the mRNA, this is the DNA sequence. And then we have the reverse complement over here, which is called the anti-gene sequence, or the reverse complement. That's the better word for it. So when we look at the way in which it is written, you can see that this reverse complement is, you will have to sort of, move the complementary strand by 180 degrees. I mean, of course, you don't do that. You learn how to write the reverse complement sequence in the correct manner. So A will base pair with T, T will base pair with A, and so on and so forth. So you're always reading this DNA sequence in a 5 to 3' direction. So when we look at the actual gene, some of the genes can be located on the plus strand, some of the genes may be located on the minus strand. So as I was mentioning yesterday, some of the strands, one of the strands is called the plus strand and the other strand is called the minus strand. In some cases, they are called the Watson strand or the Crick strand, and some genes may be located on the plus strand, some genes may be located on the minus strand. Okay, there is not much else in this particular video. I'm going to stop it and then I'm going to go to the next one, which is even more important.
Transcription.
So I'm going to let keep the voice here.
RNA polymerase makes a copy of the real gene sequence, not by reading it directly, but instead by reading the complementary anti-gene sequence. Because of the rules of complementary base pairing, the anti-gene sequence can be used as a template upon which to add complementary RNA nucleotides. The mRNA transcript that is produced is therefore a copy of the real gene sequence. A good analogy would be the use of a mold to produce copies of a production item. First, the real object is used to produce a negative imprint of the 3D shape.
So this would be the opposite strand.
Copies of the original real object are then made not by duplicating the original model, but instead by pouring new material into the mold.
So this would be your RNA.
The making of a positive shape from a negative mold is like the making of a positive mRNA transcript from a negative anti-gene sequence, where the real gene sequence is equivalent to the original object that was used to make the mold in the first place. Now that we understand the concept, we can learn the proper terminology. The DNA strand containing the real gene sequence is called the coding strand, because its matching mRNA copy will code for the corresponding protein. As we'll see in the next module, this is the code that will be used to translate from nucleotide sequence to amino acid sequence. The coding strand can also be called the sense strand because it makes sense. The opposite strand is therefore called the non-coding strand or anti-sense strand. Finally, the non-coding or anti-sense strand can also be called the template strand because this is the strand that RNA polymerase will use as a template. For the same reason, the coding or sense strand can also be called the non-template strand.
Okay. One other point to keep in mind here is that the mRNA, we are going to be talking primarily about mRNA throughout the lecture. The mRNA, and this is true for other RNA molecules as well, they're always synthesized in a 5 to 3' direction. This direction makes sense because the new nucleotide is added in a 5 to 3' direction, right? So we have to have a free 3' OH to which the new nucleotide can come, and with the phosphate, the 5' phosphate, it can form a phosphodiester bond. So the new mRNA that is being synthesized now is always synthesized in a 5 to 3' direction, and it uses the template strand, which is the complementary strand of where the gene is located. So the plus strand or the minus strand, it doesn't matter. It's the opposite strand that gives us the sequence because the sequence has to be a reverse complement of the actual gene sequence. I hope that made sense to all of you. And what we are going to do now is to have a very quick outlook again of the process of transcription. This is a very preliminary outlook, and then we'll go into more details as we progress today. So this is the very first part. I'm going to make it. So I'm going to talk here. So in the initiation part, transcription, of course, can be divided into initiation, elongation, and termination. So the idea here is to have the DNA molecule to recognize where the gene is. It's on the plus strand or on the minus strand. That will be recognized by the promoter region. So the promoter region is located on the DNA, okay? So now what will happen is RNA polymerase, with the help of many other proteins that we will look in detail today, with the help of many proteins, it's going to come and it's going to bind to this promoter in order to recognize where the gene starts. There is a nucleotide called the transcription start site, which is the beginning of the gene, and the mRNA will start getting synthesized using that particular nucleotide on the DNA as the template. But to recognize that nucleotide, the RNA polymerase needs the help of many other proteins. Once these proteins go and identify the promoter region, the RNA polymerase will come and it will bind, and with a few hiccups at the beginning, there are some, going back and forth, etc., which we will look at. The RNA polymerase then acts in a very processive manner. So here I would like you to learn this word called processive. Processive means that this RNA polymerase will continue acting as the enzyme to help synthesize the mRNA continuously. Okay, it doesn't fall off from the DNA. So it has the ability to stick to the DNA until we reach the end of the gene. Okay, remember the gene is on the DNA. The mRNA is being synthesized here, and as soon as we reach a termination sequence on the DNA, this whole transcription machinery, including this huge protein RNA polymerase, will then fall off, and we will have the RNA molecule, which in eukaryotes undergoes further processing, as we will see now. So transcription is divided into the initiation. The initiation part of it is very crucial because it helps the transcriptional machinery to go and recognize where the promoter is. The presence of the promoter sequence, and obviously it's not just one sequence, it's the presence of several sequences, which will at very specific distances from the transcription start site. So one other thing that I would like to mention here is the nucleotide of the transcription start site on the DNA is called +1, and then all the sequences upstream of that is given a number but with a negative. So -10, -35 would refer to the 10th nucleotide upstream of the transcription start site, or the 35th nucleotide upstream of the transcription start site. Okay, so this is also something that you need to keep in mind. Once the initiation happens, then this RNA polymerase undergoes certain changes in order to elongate. So this elongation process takes a little bit of time, but once it starts, it happens very quickly. So RNA polymerase moves in a very rapid and processive manner throughout the entire length of the DNA, throughout the entire coding sequence, until it reaches a termination sequence. This termination sequence is quite well established in prokaryotes. In eukaryotes, it's not very well known. Okay? So RNA polymerase and other accessory proteins bind to a DNA sequence called a promoter, which lies just upstream of the gene. Let's look at it now. So this is the huge transcription factor complex that comes and helps the RNA polymerase to go and bind. And now the RNA polymerase will bind and result in the synthesis of the mRNA. So here we have the promoter, we have many accessory proteins, these are called transcription factors, and this is the gene. So here we have the binding of the RNA polymerase, which is in blue. All the accessory proteins are coming and helping. The location and the orientation of the promoter indicates which of the two DNA strands is to be transcribed. So the promoter sequence here is important. And remember, I mentioned there has to be a sequence about 10 nucleotides upstream. There has to be a sequence about 35 nucleotides upstream. There can be sequences, a few nucleotides downstream even. And after the transcription start site, there are sequences that help in the binding of these proteins. So these, that is what defines whether the plus strand will be transcribed or the minus strand will be transcribed. And since the DNA is double-stranded and it has these hydrogen bonding between the two strands, they need to be separated, right? Because only one strand is transcribed. So this separation of the two strands is also something very interesting. 12 to 14 bases are then slightly pulled apart. The hydrogen bonds that hold them together will be broken. So this is a denaturation, a partial denaturation that happens in the DNA with the help of enzymes that are located in this hollow complex. And then we have the exposure of the specific sequence of the promoter. So here we have the separation. So this creates a transcription bubble. So during the transcription elongation, the RNA polymerase will, so this is the elongation phase now, the RNA polymerase actually moves independently of all of these proteins. These proteins are left behind. Some of them leave the complex, some of them are left behind on the DNA to mark the regions where the gene has already been transcribed. And then it proceeds along one of the two DNA strands in a 3 to 5 prime direction. So the new mRNA is synthesized in a 5 to 3 prime direction. That's something that you have to keep in mind. And it makes perfect logical sense that the mRNA is transcribed in a 5 to 3 prime direction, because that's how the phosphodiester bond is formed. As the RNA polymerase travels, it opens the transcription bubble in the front of it and closes it behind. So that denaturation, partial denaturation of the DNA will happen as this hollow enzyme, the RNA polymerase enzyme, moves forward. So let's have a look. So you can see the bubble here, and you can see the mRNA being synthesized over here. These are the nucleotides that are coming. You don't see the pyrophosphate being released, but it is also being released, which will then break down in order to give the energy needed. The sequence of the mRNA obviously is defined by the sequence of the gene. So there is a hybrid here of RNA and DNA that is formed, a temporary DNA-RNA hybrid, which is then broken down to release the RNA molecule as the synthesis happens. In the next part, we are going to look into further details, and then
initiation, elongation, and
so what we saw was the addition of the new nucleotides in a 5 to 3 prime direction. This is anti-parallel to the 3 to 5 primed template. So, of course, RNA polymerase can only polymerize the nucleotide in the 5 to 3 prime direction, because the energy, as I was mentioning a little bit earlier, the energy required to join the incoming nucleotide to the growing chain comes from the nucleotide itself, from the release of the pyrophosphate, which will then provide the energy. So this is the pyrophosphate being released here. These are the phosphate groups which are being released. And as these phosphates break down, they provide the energy. So this happens very quickly, about 60 nucleotides per second. However, this number can vary depending on many factors. And termination occurs when the RNA polymerase reaches a terminator sequence at the end of the gene. This terminator sequence is very well known for eukaryotes, but it's not very well known for prokaryotes. And as soon as it happens, the RNA polymerase leaves. In eukaryotes, this RNA will undergo a number of different processings. For example, this is the primary mRNA sequence. The primary mRNA sequence consists of introns and exons. So what are introns and exons? So this is the DNA that was being transcribed. This is the primary mRNA transcript that has been formed. This primary mRNA transcript is a faithful copy of the DNA. And what the DNA has is it has a number of intervening sequences called introns that have regulatory function, but they don't code for any gene. Exons, on the other hand, are the sequences that actually code for a gene. Okay, so here, if you look at this particular primary mRNA sequence, you can see that there are these gray regions that refer to the intronic sequences which need to be removed. Later on, we will understand why we need these introns. And these intronic regions will be removed by a process of splicing that I will explain in a little bit more detail from the slides later on. And it will also undergo a process of capping, which means that the 5' end of this mRNA will have a methylguanine cap, and the 3' end will have a poly A tail, which will then give us the mature mRNA. And this mature mRNA, only after it has been capped and tailed, only after that will it be able to leave the nuclear membrane through the nuclear membrane pore and into the cytosol to be translated. So we will now look at two more videos. The first one is the capping, the last one is the tailing, and then I will open up for some questions.
mRNA processing in eukaryotes. Capping.
So in prokaryotes, the mRNA transcript is translated as soon as it is transcribed, because it occurs in the same compartment. There is no separate nucleus, right? So this is the DNA, this is transcription happening, and the mRNA, as soon as it is transcribed, will be translated. But in eukaryotes, we have a separate nuclear membrane, and so we need to find a way for the mRNA to pass the nuclear membrane and to come into the cytosol to be translated. So they should therefore undergo a series of changes called capping, splicing, and tailing, which we are going to look at now. So one of the very first events that happens is the formation of this cap. A molecular cap is added to the 5' end of all eukaryotic mRNA transcripts. This does not happen in prokaryotes because obviously the prokaryotes do not need to cross the nuclear membrane. The cap consists of a guanosine nucleotide with a methyl group attached to atom 7 of the nitrogenous base. Normally, this methyl group doesn't exist on the guanine residue. Just for the specific guanine residue that goes and forms the cap, we will have this methyl group attached to atom 7 of the nitrogenous base. This is not on the sugar, but on the base. The 5' end of the sugar molecule, remember, the numbering is given on the basis of the carbons of the sugar residue, the ribose residue, of the, in this case, the deoxyribose. Actually, this is the ribose. Its 5' end is covalently bonded to the 5' terminal phosphate group of the first nucleotide in the mRNA. So remember, we are reading the mRNA in the 5' to 3' direction. The 5' end of this mRNA has a phosphate group, as it should. This phosphate group now will form an unusual covalent bond with the 5' phosphate of the guanosine. Normally, what we have in a phosphodiester bond is a 5' to 3' bond, right? So the 5' phosphate goes and forms a phosphodiester bond with the 3' OH. In this case, we have an unusual 5' to 5' bond that is formed. And plus this, the nitrogenous base of this particular guanosine will get methylated, a methyl group will be added, and this is called the cap. This is the methyl group, and this is the 5' to 5' bond that is formed in the head, in the cap. This is unusual, as I was mentioning, because normally nucleotides are connected in a 5' to 3' direction. Therefore, this cap is called a 5' 7-methylguanosine cap, or simply the 5' cap. Capping is a 3-step process. One phosphate is removed, guanosine is added, and then the methyl group is added. So this phosphate removal is occurring from the GTP itself. The GTP, before it can go and bind to the 5' end of the mRNA, the phosphates are removed, guanosine is added, and then methyl is added. The phosphate is removed from the mRNA, I'm sorry. Finally, we will look a little bit at splicing, and then we will look at tailing. So eukaryotic mRNA transcripts are made up of introns and exons, as I was mentioning. Exons are the regions that are involved in translation. Introns are non-coding regions that have value in regulation in the original DNA molecule. So each gene typically has many introns and exons that are interspersed with each other. So although the entire gene is transcribed, only the exons will be translated. So these intronic regions therefore need to be removed with the help of a complex called a spliceosome. So this is how it's removed. A lariat is formed, and this lariat is now removed, and then we have a phosphodiester bond formed with the mRNA. So splicing is important because it allows one gene to produce many different transcripts. So if you have a combination of various different exons, so here we can have exon 1, 2, 3 combined to form one mRNA. We can have exon 1 and 2 combined to form another mRNA, or exon 1 and 3 to combine to form another mRNA, or exon 3 alone to form a fourth mRNA. So from one gene sequence, we can actually get 4 different mRNAs, right? So this allows us to have many more proteins than we have genes. So the human genome has approximately 21,000 genes, but because of splicing, it has many more proteins, more than 50,000 proteins. Finally, we will talk about the poly-A tail, and then I will open up for questions. This is the last processing that the mRNA goes through, which is called addition of the poly-A tail. This is a chain of approximately 200 adenosine nucleotides, only A's, no other nucleotide is added. That is added to the 3' end of this mRNA. So here you can see this is a poly-A polymerase, which goes and adds the adenine nucleotides to the end of the mRNA. Several accessory proteins can bind to the mRNA transcript before it can exit the nucleus. So here on the poly-A tail, you can see that many proteins are binding. These proteins can help prevent the mRNA from folding onto itself. So it will prevent it from forming these higher order structure in regions of self-complementarity. And this will allow the macromolecule to remain single-stranded, not fold too much, and exit the nuclear pore in an effective manner. So there are also other accessory proteins like PABP, poly-A binding protein, that can bind to the poly-A tail to promote nuclear exit. Only once the mRNA has undergone all 4 of these modifications, only then it can exit the nucleus. So here I'm going to talk about something very interesting, and that is a process of quality control. For an mRNA to exit and go to the cytosol and then be converted, then be used as a template for protein synthesis or the translation process to happen, the mRNA needs to be absolutely accurate, right? It should not have any mistakes in it. So for example, if the splicing has not happened properly, if the capping has not happened properly, if the tailing has not happened properly, if these processes have not occurred properly, then this mRNA is not going to exit the nuclear pore complex. Even if it exits the nuclear pore complex, it will be, for one, it doesn't exit. If it does, it will not be able to go and bind to the ribosome in a proper manner in order to be able to start the translation process. We will learn that in our next lectures, that the capping and tailing is also essential for this mRNA. Remember, we are only talking about eukaryotic mRNAs here. It can only exit the nuclear pore in this manner, and so it undergoes a number of quality control checks before it can be it can exit the nuclear pore and before it can be used for mRNA translation. So so it will go and bind to the ribosome, and then translation will start. So let's have a look at this as well. So fully processed mRNA will have the 5' cap. It will have the 5' untranslated region. This is also very crucial. I'm going to talk about it a little bit. We will have the coding sequence, and then we will have the 3' UTR, and then we will have a poly A tail. So what does the UTR mean? The UTR is the untranslated region. So this mRNA is going to now go to the cytosol, and it will go and bind to the ribosomes, and then it will undergo translation. For this translation to happen, this translation will happen only within the coding sequence. But upstream and downstream of the coding sequences, we have these untranslated regions called UTRs, which are absolutely essential for the regulation of translation of this mRNA. So we will learn this as well in a later lecture, how the transcription process of transcription and translation both are regulated very, very closely. During the regulation of translation, this mRNA can actually have several different RNA molecules like microRNAs or siRNAs, as well as RNA binding proteins that can go and bind to the untranslated regions of this mRNA and affect whether this mRNA can be translated into the protein or not. So this 5' UTR and 3' UTR, the UTR regions can be very long, they can be variable. One same mRNA can have a short UTR, it can have a long UTR. If it has a longer UTR, it can have many more regulatory proteins and miRNA, siRNAs binding to them. So the length of the UTR is also important. And then we have a poly A tail. At the poly A tail, there isn't much regulation happening. The poly A tail is just necessary to establish the end of the mRNA molecule. And then we have the poly A tail after that. One other interesting thing that you may notice in this mature mRNA, this is called the mature mRNA now, because it has the cap, it has the tail, it has been spliced, is that at every single splice site, which has not been represented in this picture very well, but what these little dots mean is that at every single spliced site, there are some proteins that go and bind in order to give the idea that this particular protein, this particular mRNA has been spliced accurately. It has undergone a proper splicing process. So at each specific splice site, some proteins will go and bind, and these proteins will be stripped off, they will be removed while the mRNA is making its way through the nuclear pore and into the cytosol. This stripping of the proteins that are binding to these intron-exon junction regions, these are called junctional proteins, the stripping off of this protein is also a quality control measure. If these are not stripped off properly, or if there aren't enough intron-exon junction proteins available suggesting accurate splicing, then this mRNA will be degraded before it can be translated into a protein, because obviously the protein that will be formed will be meaningless if the mRNA sequence is not correct. So this is another quality control measure that we have in place. So the UTRs and the coding sequence represent all the exons that were spliced together. The 5' end protects the 5' end of the mRNA from enzymatic degradation. That's also something very important. Why we need this cap is because we have a number of nucleases that are found in the cytosol. These nucleases can be RNases, they can be DNases, but the RNases, the nucleases, they can degrade the mRNA. So this mRNA should not be degraded until it has been translated. So the presence of the cap suggests that this mRNA has not been translated yet. It needs to be translated, and then the cap is removed and this mRNA can be degraded. It assists in ribosome binding as well, as we will see later, and the coding sequence will be translated into the protein. The 3' UTR is important for translation termination and for post-transcriptional modification, which is via the miRNAs. And the poly-A tail prevents or delays the enzymatic degradation of the 3' end of the mRNA. So 5' end degradation is prevented by the cap. 3' end degradation is prevented by the tail. The poly-A tail can shorten as the mRNA gets older, and by older I mean we measure mRNA age by minutes rather than any other unit of time. So as this mRNA gets older, its poly-A tail length decreases. If the poly-A tail length decreases to a critical limit, it means that this mRNA is old enough now, it has lived its life, and it will be degraded in specific little structures that we find inside our cytoplasms called P bodies, the letter P bodies. So P bodies will take in the mRNA, and nucleases in those P bodies will actually degrade the mRNA into the nucleotides, and these nucleotides are then going to be recycled. So UTRs should not be confused with introns. So although UTRs and introns are not translated into the protein, UTRs are found in exons. So it is a part of the exon of the coding sequence of the DNA. The UTR is a part of it, but when it comes to the mRNA, it will not be translated into a protein. It's not included in the finally. The UTRs are found and they are included in the final fully processed mRNA, but the introns are not. Okay. So now I am going to stop my sharing, and I will ask if you have any